Hello, everyone. My name is Joseph McClay. I'm an associate professor at the VCU School of Pharmacy. My background is in psychiatric genetics. I did my bachelor's and PhD in the United Kingdom before coming to do a couple of different postdocs at VCU, looking at genetic risk factors for psychiatric disorders, and then also psychiatric pharmacogenetics, or looking at how genetic variation affects response to psychiatric drugs. My professional experience, I've had a variety of different positions at the VCU School of Pharmacy. I'm currently an associate professor, as I mentioned, and I joined the Center for Pharmaceutical Engineering and Sciences in 2021 as a faculty mentor for a summer student as part of the National Science Foundation Research Experiences for Undergraduates grant that was received by the center. So the focus of my lab is functional genomics and epigenomics of central nervous system drug response. And what this means, we basically look at ways to potentially drug the genome to change how genes are regulated so that that can potentially alleviate psychiatric symptoms. And also we look at how variation between individuals in their genetic regulation affects how they respond to drugs. One of the major techniques that we use is what's known as next generation DNA sequencing. And this technique has been facilitated by the vast improvements in technology and reduction in costs of sequencing genomes over the last couple of decades, as you can see from this graph. So one of the methods that one of the things that we're focusing on right now is drugging the genome to treat psychiatric disorders. And this is a potentially novel thing because, well, we do know that for sure now that there's a substantial genetic component to risk for psychiatric disorders. And what we're trying to do is target that genetic component to change the regulation of risk genes as an alternative to the existing drugs, which are often not very effective. So the genome itself does not exist in isolation in the cell. It's sort of a complex thing that's represented here by this black line, but it's wrapped around these proteins called histones. And the genome can undergo all sorts of different modifications, epigenetic modifications, to mean that it's either switched on and it's this open state where it's accessible to different proteins, or it's closed off and shut down in this state here where the histones are sort of pushed close together. And one of the things that regulates the change with these two states is what's known as histone acetylation, or the addition of acyl groups to the histones. And when there's these acyl groups there, then the chromatin unwinds and is accessible. And we've been experimenting with a novel compound that was synthesized by a group in the Midwest a couple of years ago that targets a specific histone acyl transferase enzyme. And we've shown that we can actually change the regulatory state in human cells, human neuroblast cells, around the HPF1 schizophrenia risk gene. We can change the degree of histone acetylation around there. So this is just one small example. We've had a lot of funding over the years in looking at transcriptional regulation in psychiatric disorders. And now we're really trying towards trying to modulate this using drugs. So I think this is quite an exciting area. Another area we're looking at is epigenomics of drug response in aging. We have current NIH funding to do this. This shows some work done by a recent graduate from my lab, where basically we show that DNA methylation, which is another epigenetic marker, increases with age at a certain gene that encodes an enzyme called cytochrome P452E1, which metabolizes drugs. So the increase in DNA methylation results in a decrease in gene expression with age and a decrease in the expression of the enzyme, and that changes how rapidly the drugs are metabolized. And this has important relevance for biomarkers potentially in older individuals who are much more at risk for adverse drug reactions. So trying to use biomarkers to modulate dose in the elderly. Past graduate students, I really only have the one guy who's already graduated, this guy, Mohammed Kronfall. After he left, he did about a one-year postdoc at Johns Hopkins before moving on to full-time employment at the Food and Drug Administration in the Washington, D.C. area. I have two current PhD students, one in their final year and one in their third year. And I've mentored a variety of different students on their advisory committees, as well as PharmD students, undergraduate students, and so on. And I'd be happy to take your questions about projects in the lab later on. Thank you.